Welcome back to Everyday Biology. If you've ever downloaded DNA or protein sequences from the internet, you've probably seen weird-looking files called FASTA or GenBank formats. Today, we'll break down what these formats mean, why they matter, and how to read them, all in a simple and beginner-friendly way. Before we jump into FASTA and GenBank, let's understand what a sequence format actually is. When you retrieve biological sequences like DNA, RNA, or proteins from databases like NCBI or Ensemble, they come in standard text formats. Why? Because these formats help computers store, read, and share the biological data in an organized way. Think of it like recipes. The ingredients, nucleotides, or amino acids are listed, but you also need the title, source, and instructions. Similarly, sequence formats contain both the sequence data and the metadata. Let's start with the most common format, the FASTA format. 1. It's super simple and widely used. 2. You'll often see files ending with .FASTA, .FA, or .FNA for nucleotides, or FAA for proteins. A FASTA file has two parts. 1. A header line that starts with a greater than sign. 2. The sequence, written in single-letter codes. Here's an example of a DNA FASTA file. What does this mean? The line starting with is the header. It includes an identifier and a description. The next lines are the actual DNA sequence. FASTA is great because it's easy to read, compact, compatible with almost all bioinformatics tools, but it has limitations. It doesn't store detailed information like gene location, references, or features like exons and introns. Now let's talk about the GenBank format, developed by NCBI. It's a lot more detailed than FASTA and contains rich annotations. GenBank files usually have the extension .gb or .gbk. Here's what a GenBank file contains. 1. Locus, the ID, length, molecule type, DNA or protein, and source. 2. Definition, a brief description. 3. Accession, unique identifier. 4. Features, this is the key part. It includes CDS, coding sequences, exons, introns, promoters, regulatory regions. 5. Origin, the actual sequence at the end. Example, simplified version. Key point, GenBank format is essential when 1. You need detailed annotations. 2. You're analyzing gene structure. 3. You're doing comparative genomics. But it's heavier and not as easy to parse with simple tools as FASTA. Use FASTA for basic sequence processing and alignment. Use GenBank when you need annotations and feature data. That's it for today's episode of Everyday Biology. Now you know what FASTA and GenBank formats are, how they differ, and when to use which one. If you found this helpful, don't forget to like, subscribe, and share with your fellow biology nerds. See you in the next video, where we'll dive deeper into sequence analysis tools. Stay curious.